Good afternoon. I'm very glad to have the opportunity to present the work I've done during my postdoc at the Columbia University in the group of Gordana Vunjak-Novakovic in collaboration with University Hospital Basel in the group of Andrea Banfi, where I'm actually working now. Following myocardial infarction, there is the formation of a collagenous and stiff scar that impairs the cardiac functionality, as everybody knows. And cardiac tissue engineering has been envisioned as one promising approach in order to revert the sequelae of myocardial infarction and to repristinate the cardiac functionality, contractility. Our aim was to generate a cardiac patch in order to loaded with contractile cells to be implanted on the top of an infarcted area in order to actually help to repristinate the cardiac functionality. In our group, we were focusing on the use of a perfusion culture system in order to allow the engineering in vitro of a thick cardiac patch, millimeter thick cardiac patch. What we used at the Columbia University was the use of neonatal rat cardiomyocytes combined with an elastomeric scaffold, polyglycerosebacate. In particular, this scaffold had the characterization of multiple channels that should mimic the vascularization, the capillary network typical of the native myocardium. Indeed, by the perfusion of culture medium through the channels, we should have overcome the oxygen limit diffusion of 100 micron. So as I said, we were using a perfusion bioreactor system, both for the seeding and the culture of cardiomyocytes on the polyglycerosebacate. This is an example of the application of these methods, in this case on a more simple model of a single central channel. Here we investigated the stiffness, the most appropriate stiffness of the polyglycerosebacate to be used in order to obtain a contractile patch at the end. And as you can see, we were able at the end to have a quite functional patch, cardiac patch at the end. But the problem is that how we could guarantee the survival of such a cardiac patch upon implantation. So still there was an open issue of the vascularization once in vivo. And here we thought to combine the cardiac tissue engineering approach with the cell-based gene therapy. And we collaborated with Andrea Banfi. Here we use the concept of the use of cell delivering VEGF. And as we already know that VEGF is one of the most potent angiogenic factors, but still we really need to control the microenvironmental dose of VEGF. Andrea Banfi previously showed in a clonal study that the transduction of skeletal myoblasts that express a specific level of VEGF brings to a range of safe and therapeutic VEGF levels that brings to a deformation induction of normal angiogenesis. Below and above this range, we have either an inefficient angiogenesis or the formation of aberrant structure. So this study has been done in a clonal population, but in order to, it's quite difficult to work on clones. So recently, in Andreas Banfi lab, they developed a FACS technique that allows to purify a population of cells expressing an homogeneous level of VEGF. What we did, it was to retrovirally transduce the cells with a construct where VEGF, rat VEGF, was directly linked to the truncated version of CD8. This allowed to have these two genes encoded in the same messenger RNA which brings to a strict relationship among the expression of CD8 at the membrane of the cells with the VEGF released in the microenvironment. So we were able, by FACS sorting, to sort a specific expression of CD8 at the surface marker of the cells, and therefore to have cells expressing an homogeneous level of VEGF, which is in the same safe and therapeutic level window that has been shown before. So, as I said, in this study, we tried to combine the cardiac tissue engineering with the cell-based gene therapy in order to generate a cardiac patch, a contractile cardiac patch that has the intrinsic angiogenic potential. We tested the angiogenic, we used the same methods developed before, but this time we co-cultured the neonatal rat cardiomyocytes with FACS-purified skeletal myoblasts that expressed an homogeneous level, a homogeneous and safe level of VEGF. We tested the angiogenic potential of these cardiac patches in a nude mouse ischemic model. We performed the infarction by the ligation of the left anterior descendant artery. 1 week after the infarction, we implanted either the scaffold alone, PGS, polyglycerosebacate, or patches generated by rat cardiomyocytes alone or co-cultured with skeletal myoblasts expressing truncated version of CD8 as a control or expressing an homogeneous level of VEGF. Then after 4 weeks, we sacrificed the animals. As the readout for the cardiac functionality was to perform a high-resolution echocardiography, which has been performed at day 0 before the infarction, 1 week after the infarction in order to assess the quality of the infarction, and 4 weeks post-treatment. Here, the high-resolution echocardiography allowed us to calculate in the area of interest, so in the area where the patch has been implanted, to calculate, to measure the strain. And here in the graph, you can see calculated both short and long axes, the strain fold increase of the echo performed at 4 weeks, normalized to 1 week echocardiography. And as you can see, when the VEGF patches has been used, the functionality, the cardiac functionality improved quite a lot as compared to all the controls. So we tried to investigate the reasons behind this higher functionality. And what we observed is that only in the presence of the VEGF expressing patches, the patches formed a quite compact and dense tissue that has been well integrated with the surrounding tissue, whereas all other controls formed quite a loose and less compact tissue. We analyzed the thickness of the different regions of the myocardial wall. And we, in particular, we quantified the thickness of the infarction, the patch, and the myocardium. And what we observed is that at the infarction, the thickness of the infarction is quite similar in all the experimental conditions. This brought us to conclude that our patch, VEGF expressing patch, did not affect really the scar formed after the infarction. But when we went to have a look to the thickness of the patch, we see that the thickness of the patch itself has been maintained in the 4 weeks and is the highest as compared to all the other groups. The myocardium, and surprisingly, we observed that the thickness of the myocardium calculated at the border of the infarction, so at the border also of the patch where has been implanted the patch, the thickness was much higher as compared to all the other groups. And this has been clearly explained by the fact that only in the VEGF expressing patches we had the survival of the loaded cardiomyocytes, and as you can see, the cells have been also an elongated morphology and seemed to be morphologically interconnected among them. And by XGAL, we were able to identify also the skeletal myoblast that express in VEGF that has been previously retrovirally transduced for beta-GAL. And here we see that only in the VEGF-treated patches we have the survival of the skeletal myoblast, and not only, they were also able to differentiate into mature myofibers. And this is well explained by the fact that the quantification, the vessel quantification was higher in the patch treated condition, much higher the vessel density as compared to all the other controls. And as we can see here, the morphology of the capillary formed by the VEGF expression is quite normal and is well surrounded by the pericytes in green and G2 positive. Surprisingly, we found out that the vessel density increased also in the VEGF-treated patch, also in the myocardium surrounding the patch and the infarction. So, in conclusion, these results show the proof of principle that controlled VEGF expression by FACS-purified transduced skeletal myoblast effectively increases the vascularization of the patch and the myocardium. This shows that we were able to generate indeed a patch with not only an intrinsic but also an extrinsic angiogenic potential. This brought to a better survival of the loaded cells and therefore a better thickness of the myocardial wall. And we also observed the cardiac function improved as a consequence of either or both the previous two mechanisms. Of course, this proof of principle we obtained by using skeletal myoblast and neonatal rat cardiomyocytes, which are not clinically relevant cell sources. So next step, it will be to substitute the skeletal myoblast with other clinically relevant cell sources, as we already started to FACS-purify transduced mesenchymal stromal cells. And next step will be also to substitute the rat cardiomyocytes with other sources of cardiac progenitors, like inducible pluripotent stem cells or embryonic stem cells. So I want to thank everybody that contributed to this study and all of you for your attention.